Hi, my name is Nashwa. So today I'll be talking about my project on photobiomodulation enhancing muscle performance in mice. I have no disclosures. The project has two aims. Aim 1 is based on cells. I used aim 1 to determine which wavelength is necessary for performance analysis. My model was using C2, C12 myotubes, and the wavelengths I looked at were 980 and 810 nanometers. In this aim, I had electric stimulation where I can stimulate my cells to contract, and I looked at two wavelengths where 810 nanometer was the most suitable, and I chose that to apply on my aim 2, which was in vivo, looking at mice. Today, I'm going to show results only for aim 2. The assays I used for aim 2 was to look at a treadmill uphill run, and I induced fatigue, so I have like a fatigue protocol where the mice can't run anymore at a certain point. What I saw was that my PBM mice ran double the distance than controls, and this is possibly due to activation of regeneration occurring in the muscle. So for in vitro, the PBM device I used is made by Alexa, who is an engineer in our lab. She engineered this device where I placed my cell culture plates on the top of the device, so on here. So at the bottom of each circular base, there is an LED. She made a 980 nanometer LED device as well as an 810. The picture here is a purple LED board that has 980 nanometer LEDs on it. For in vivo studies, I used an Omnilux panel, which is an 830 nanometer wavelength, and I placed my mice underneath the panel. These mice are anesthetized because the exposure time was about 20 minutes. After exposure to PBM, I placed the mice on a treadmill, and I do a fatigue test. So in a fatigue test, you angle the treadmill to about 10 degrees uphill. You also have a progressive increase in speed. So the starting speed was 8 minutes per minute, and the mice ran for maybe 1 minute at that speed. Eventually, near the end, the maximum speed they run is 26 meters per minute. And I measure time to exhaustion, so how long they ran is how I measure their fatigue. So this is a video, and I'm not sure how to play it, but basically just on the picture you can see that the control is running near the bottom, which is the shock grid. We use shock for a negative reinforcement for them to run. The treated mice, they run higher up the lane. So the point of exhaustion is defined if the animal is spending about 10 seconds or longer near the shock grid. So the control at this point is fatigued. So I would take out the control from the treadmill, note the time of exhaustion, as well as measure the blood lactate. What I see is that all my controls, they run about 20 minutes. And the treated ones, they run twice as long, which is especially, you can appreciate that more at the 24-hour post-PBM treatment. For lactate, there didn't seem to be much of a difference between control and PBM for the ones that ran less than 40 minutes, but mice that ran longer, which were only the PBM treated ones, had a decline in lactate levels. We then, after their run, we took their muscles and did RNA sequencing analysis, and we saw a number of genes that are involved in tissue remodeling, muscle morphogenesis. So we did some histological analysis, and we saw that in controls there is some damage where the dystrophin, which stains the fiber nicely around the structure, is aggregated. But in the PBM, the natural structure of the fiber is retained. We did some organelle imaging, so looked at mitochondria. What we saw that in controls, the mitochondria is less dense, so more gray looking, but in PBM, the mitochondria is more darker, dense, suggesting that the mitochondrial morphology was protected. So in conclusion, with the performance, we see that the PBM-treated mice run longer, possibly due to activation of regeneration going on in the muscle. There could be potential infiltration of other cell types, helping to attenuate injury and fatigue muscles. So I would like to thank Rox Anderson and the team, and Josh, as well as the other members in the Anderson lab. Thank you.